Greetings. It's so nice to see the pictures or names of fellow microbiologists around the world, that we are united in our love for microbiology. I've been an ASM member for over 50 years, and it's so wonderful to connect through Zoom that Kirsten has engineered here with you about the information I want to share. And I'm going to be, in my presentation, Water Quality Testing at the Community Level, I'm not just going to be presenting. What I want, the outcome of it is, after you see the things that I am presenting here, that you could see, hey, I could do that myself. And for some of you who have waterborne disease as a major problem in your countries, you could do these things. And I could maybe help you do those things. And at the end of my presentation, I'll put in my email address so that I can connect with you. And let's see if microbiologists together can address waterborne disease. So greetings, and I'll start off. Let's see here. Okay, how do I advance it?
Sure. There's an arrow on the bottom left hand of your slide on the screen there. Other
Okay.
side, the other direction.
Oh, okay.
There you go.
There you go. Okay. And I don't have any financial connection with the companies whose products I'll be talking about here. So just that disclosure. But I call waterborne disease an overlooked chronic epidemic. And some of these fact sheets say that today, there'd be 4.5 million cases of diarrhea and over 1,200 deaths from children, under 5 years of age. And, 6 million 6 million cases of diarrhea worldwide. This is a major problem, okay? And a lot of people will talk about the statistics. 2.1 billion people lack safe water at home. But these are not statistics. These are real people. This is in Kisii, Kenya. How safe do you think that would be to drink the water? Or this is in Mwanhuzi, Tanzania, collecting water from a dry riverbed. And this is Dar es Salaam in Tanzania, collecting water there. This little girl with that heavy thing on her head. And the United Nations General Assembly, they declared safe and clean drinking water a human right, essential for the full enjoyment of life, back in 2010. But those are empty words. All your countries that signed this, what are they doing in the last 15 years? And as we know, as microbiologists, left out of the UN General Assembly resolution was the right of all people to test their drinking water sources to determine the disease risk. And without microbiology testing, citizens are clueless about the safety of their drinking water, and governments can't be held accountable. And what I will be showing you then is that the portable microbiology laboratory, that will fix this. Okay. And waterborne diseases are preventable. Kill the germs with chlorine or heat, and people don't get sick. That's simple. And of course, microbiology is the key to understanding waterborne disease. But microbiology is difficult to understand for most people then, because the bacteria are so small. Look at this, thousands of bacteria can fit on the tip of a pin. And how can tiny bacteria cause animal disease? And that's because we know, and we always teach, their exponential growth. 10 doublings to get 1,000, 10 more to get 1,000,000, 10 more to get 1,000,000,000 in a nutrient broth culture or on a colony. So the key concepts are that every 10 generations, there's an increase of 1,000 in the in the population from 1,000 to 1,000,000 to 1,000,000,000 then. And to see evidence of bacterial growth, at least 1,000,000 cells are needed. That's what you're going to, and that requires one cell to grow 20 generations on a petri dish, so you can start to see a colony. And at 30 minutes for each generation after a lag phase, it'll take about 10 hours for one cell to become 1,000,000 cells and for tests to become positive then. And of course, as we know, the microbial world was invisible to everyone until 1676, when our champion, the first microbiologist, Antony van Leeuwenhoek, made these tiny microscopes with magnifying lenses in it that can get 200, 300 magnifications. And he recorded then what we can see in a fusion today. He could see bacteria. So that was 1676, but people didn't think that these tiny things could have any effect on them. And it wasn't until 1876 that Robert Koch developed the methods then. And there's this wonderful book, Germ Theory, that ASM put out that kind of documents how we go from ignorance to knowledge. And another one of our heroes is Robert Koch. And he was the one then that gave the methods to isolate bacteria in pure culture and to see eventually if it could cause disease. He proved that with anthrax. But then using his methods, the bacteria causing major waterborne diseases were discovered. 1880, typhoid. 1883, Koch himself demonstrated cholera was a bacterium, and then Shigella dysentery. So in the 1880s, it's understood that bacteria from feces could contaminate water and cause disease. But how do we test water? And it was not practical to test for all of those, and the methods weren't very good there. Instead, there was a search for a bacterium that would indicate recent fecal contamination. And what's the job description of this bacterium? Well, it always has to be present in large numbers in the feces of humans and mammals. And a key thing is it doesn't multiply once it leaves the body and enters the water. But then, unlike bacteroides, it needs to survive at least as long as bacterial pathogens and then be relatively easy to detect by inexpensive methods. And so for over 100 years, the most specific and reliable indicator is our friend Escherichia coli. And therefore, it's recommended as the indicator of choice. E. coli will give us then, if present, recent fecal contamination. And here's a picture of Theodore Escherich, who isolated, the first to isolate then E. coli from feces of a child. And we get his name then is the E of E. coli, all right? And I'm an old microbiologist, over 15 years an ASM member. And the early years of teaching at California State University, Sacramento, we had these horrible tests that were not specific for E. coli, and they were lactose-based tests. And so in the 70s and 80s, I had to do these MPN procedures, and particularly this thermal tolerant coliform test. Oh my gosh. And it wasn't specific for E. coli because it's lactose-based. But the discovery that E. coli produced a specific enzyme, the beta-glucuronidase, not present in environmental coliform bacteria, enabled a new generation of tests to be developed specific for E. coli in the 1980s and 1990s. And on the left is Stephen Edberg, and he was a clinical microbiologist at Yale University, and he's the one that came up with this new test then. It used to be manual typewriter microbiology, but Edberg came up with this test called Coli Alert that then was like a computer replacing a manual typewriter. And this is the test which is used worldwide. And it's also what we would like because WHO had guidelines. And there's a low disease risk if there's no E. coli in 10 mil. So wouldn't it be nice if there were a 10 mil test ready to inoculate? And that's what came out in 1988. And this is the Coli Alert test. And it's called a defined substrate medium because it has two substrates in it that bacteria could use as energy sources, and then just inorganic nitrogen, sulfur, and phosphorus. So they're going to have to make all their amino acids, vitamins, purines, pyrimidines, and we know that E. coli can do that, all right? And the two substrates in E. coli for growth in this defined medium, one is ONPG. G is the sugar. And all coliform bacteria, environmental coliform and E. coli, have the enzyme that can split off the G, which they can use to generate ATP, make their monomers, put them together as polymers, and grow. And so if the medium Coli Alert turns yellow, that means you have bacteria that have that beta glucosidase enzyme with it. But then there's this second substrate in there, MUG. There's the G, and E. coli is the one that has the glucuronidase that splits off the G, and it uses that for energy to grow with. And so first Coli Alert came out as a 10 mil test, but in the United States and other places, you got to test 100 mil. So around the world, there are perhaps millions of tests today using the 100 mil, and you have a snap pack to add the same reagent, the Coli Alert powder, to this, mix it up, and incubate it overnight. But we're interested particularly in that 10 mil test because this is what you could take. If you had this, you could take it, and this is in Temeke Municipality in Dar es Salaam. You're going to collect in a whirl pack, and then you're going to inoculate. You carefully take off the cap. You use the sterile pipette to add 10 mil to the Coli Alert tube, put the cap back on, mix it up, dissolve the nutrients. And then if you have an incubator, you can put it in an incubator, as my friend John Rimbiria in Embu, Kenya has. But if you don't, you're out in the field and you happen to be alive, you can use your body. And I put it then close to my body then in a sack like that. And then my friend Faustine in Kenya, she can incubate things close to her heart then. And overnight, you get the results. The bacteria are going to grow from the one cell, perhaps, into tens of millions of cells, and you get color changes. All right? And so if there are treated water, you're going to have clear, boring microbiology. But if the water is not treated, even from a protected borehole, it's going to be yellow because the environmental coliforms, Enterobacter, Klebsiella, Citrobacter, they grow out in the environment. They aren't feces indicated. So it'll turn yellow. And then you use a UV light to shine on these things and to find if that MUG substrate has been utilized by E. coli bacteria, releasing the MU, which will fluoresce. And this one has E. coli, and this one over here is going to be a low disease risk, but not this one. It's got E. coli in it, okay? And I had the privilege then of meeting this wonderful woman, Wangari Maathai, Nobel Peace Prize winner. She has a PhD in zoology, and showing her these tests then. And she understands substrates and enzymes then. And anyone in Kenya will recognize her and what a privilege I had to show her that microbiology could be done easily there in Kenya. So with a 10 mil test, we have the, it's a presence absence test. If E. coli is present, it's not low. But if it's absent, it's a low disease risk. Well, it would be nice also to have the high disease risk is 1 to 10 E. coli in just 1 mil, and a very high, more than 10 in 1 mil. And wouldn't it be nice if there were a simple, ready to inoculate test for E. coli that we could use to find out the quantity of E. coli there, to identify high and very high risk. So how do we perform a quantitative test for E. coli? And that's where I was a food microbiologist, and I did so many plate counts of dilutions of food. You'd add 1 mil dilution, you'd pour in the agar, and you count it. But the brilliant people at the 3M company in the United States came up with a Petri film. And their thinking was, well, we can just put some dried nutrients on a piece of cardboard, and then we'll cover it with a film, and people could add the same 1 mil to this and incubate it, and they wouldn't have to use Petri dishes. They wouldn't have to use autoclaves. Oh, that transformed food testing then in 1994. That's when it first came out. And so here it is, the Petri film that they were looking for E. coli. And I'm using the Petri film then that has the nutrients for E. coli in it. So you're going to inoculate this and put the information on it. And then you're going to take just 1 mil, okay? You're going to lift up the film. You're going to add 1 mil directly to this. You're going to slowly lower the film down. And then you're going to use a plastic spreader, or you can pick it up to distribute the 1 mil over the surface. And then they're ready to incubate. But they just have 1 cell. And so you're not going to see anything until they grow into millions of cells. And so I put them between 2 pieces of thick, firm cardboard. I incubate that on the small of my back. I sleep on it at night. And then overnight, you've got the results. And the results, E. coli will be a blue colony because the glucuronidase substrate, the glucuronidase enzyme has cleaved the G off of a different glucuronidase indicator, BCIG. And so it's going to be blue because the BCI doesn't diffuse. And then there is a gas bubble around that because this medium which is used is violet red bile agar. It has lactose in it. So, oh my gosh, a blue colony with gas bubble, that's E. coli. So you can count them up here. Now the red colonies with gas bubbles, those are other coliforms. Klebsiella, Enterobacter, Citrobacter, they ferment lactose, but they don't metabolize the BCIG. So they will have that. And then other red colonies without gas, they're just environmental gram negatives that are there. So, oh, we've got a test then. So, and you explain that to people, and we take a look at these 2 Petri films. And we're looking for the Petri film where E. coli has grown in a blue colony and a gas bubble. And we take a look at the one on the left here, and that might be from a borehole, a good borehole. There are none in that. And the coli alert tube would have been yellow, but it would not have fluoresced. It has these environmental coliforms in it, but it doesn't have E. coli. And the one on the right then, oh my gosh, that's E. coli. So it's easy to interpret these tests then. And so for high and very high disease risks, this Petri film is perfect for determining those levels. And so let's just, as I tell my people here, or This is the people in Kenya. Let's put these tests for thermotolerant coliforms into a museum, and let's use the lactose-based test, and let's use these glucuronidase-based tests and enable people everywhere to do simple microbiology. So here, this is in the lab in Nairobi then, where they were setting up MPNs there, 3-tube MPNs. No, let's replace them with coli alert and Petri films here. So these are the things that you need. You need a coli alert, you need a collecting bag, sterile collecting bag, and you need a pipette to inoculate the 10 mil coli alert tube and a UV, oops. All right, let's see. All right, let's see. And a UV light for the coli alert test. You need a Petri film, need a spreader, or really you can just tilt it to spread it around. And I put it together. And this is a portable microbiology laboratory that has coli alert, 25 coli alert tests, 25 Petri film tests, 25 pipettes, and flip it around and it has 25 whirl packs in it. And you can go any place, anytime to do water testing with this, you know. Testing in developing countries. You've got this, and then you can inoculate anywhere, anytime, and anyone, incubate overnight, and then you get the results the next day. So this is what makes microbiology possible. And for the UN Habitat that I worked with a couple years, and I also made this booklet about a practical method of water testing that I'd be glad to send you the link to. And let me just show you, and this was over 20 years ago. Let's go to Sakasaka village in Shinyanga region, Tanzania. And this could be you in your villages, all right? And this is Sakasaka village. And instead of me that needs Boniface as my translator, you could be out there with a local language and you wouldn't need to have a translator, but you're going to be doing the same things. And I was with my former student, Christine Polinelli from Australia then. There are 2 water sources in Sakasaka village, okay? And one was from a pump, but you have to pay a few shillings to get 20 liters of that, and people are impoverished. The alternative then is this water from the stream, which often is clear and it's free. Now you and I, all you microbiologists would say, hey, I will find a few pennies and get the water from the pump. But they have no idea of what the situation is. So let's educate the community, all right? So you are going to test the stream and you'll test the pump. And you'll test the stream and others will test the pump. So we got about 20 people, 10 each doing the pump and 10 each doing the stream. So we show people how to get the water and inoculate the coli alert tubes to mix up like that. And then you do the Petri films and you show the technique with it. And then they can do these tests. These people that have had no science background can do the tests. And then the next morning, we set up these simple cook-it solar cookers that I'll talk about later. And one of them is pasteurizing water, and the other then is cooking rice, all right? And Boniface is translating, and you got the community out there. This is really remarkable of what's happening here, because the people have incubated overnight the coli alert and Petri film tests. So let's use a UV light, and we have to have a dark thing to see if it fluoresces or not. And the tube from the stream fluoresced. The tube from the pump was yellow, but it didn't fluoresce. And so it didn't have E. coli in 10 mil. And then let's take a look at the Petri films. And we take a look at these, And we'll line these up then. These are the tests that the community has done. And this is a wonderful example of what you could be doing places then where people have no idea about the germ theory of disease. These first 4 have lots of red colonies on it, maybe about 100. That's from feces, those blue colonies. And then that's from the stream water. And then from the pump, they're red colonies, but no E. coli. So we go around and show people then, oh my gosh, evidence-based microbiology, that the water from the stream is contaminated with fecal microbes, but not the pump. And then, oh, we've also done solar cooking. So here we have then and I leave with them materials, the coli alert and Petrifilm tests, so that they can continue to monitor their things. And you have this whole group then that's been educated. They understand waterborne disease now. They've done the tests themselves, how proud they are that they can tell neighbors then that we've tested the water and stay away from that water from the stream. You've got to find those shillings to buy the pump water. And I also had the privilege with Boniface again as my translator to have chiefs and village health workers for 17 villages that came to a workshop that I have. And I explain all about bacteria. And here you got the chief. He's doing coli alert and Petrifilm testing. And the next day results, we can take a look at that. And the chief says that has E. coli in it from the riverbed water. And we can't be drinking that. We need to treat that water. And then you take a look here, and there are lots of E. coli colonies there that they understand the water is unsafe to drink. It's a high disease risk. And then we've given each of the villages then the portable microbiology laboratory they can take back to their villages and do those explanations to their village people. And when they understand that it's water from gets contaminated from feces, that ends the open defecation, and people can start to take steps to do things. And then I worked with the Temeki municipality in Dar es Salaam, and this is Mr. Muna, who's the chief health officer, and provided them with hundreds of coli alert and Petrifilm tests then. And that this providing with lots of tests, that's Mr. Muna again, and it's my friend Safe Sweaty, who has collected the sample and does all the testing for that. And there is a letter then about what is the effect of that. And if we take a look at the diarrhea, in the year before they did testing, you got over 1,400 or so. And then with the testing that they could do in the community and identify waters that were contaminated and then give people WaterGuard chlorine to test it, look at that drop. That could happen everywhere then. And I had UN Habitat workshops for the community of Bondo, explaining again to people from all types of backgrounds about bacteria, their size, how they grow. We're looking for an indicator. Its name is E. coli. And then the second day, they understand the microbiology of water testing. I've had the opportunity to also educate a community in the Philippines. And then in Ethiopia, again with UN Habitat, this is what you could do if you wanted to organize things. You could have all these things that you talk about in the workshop and and have people do the water quality tests. I had a USAID integrated health project in the Democratic Republic of the Congo. Of course, USAID projects everywhere have been obliterated. And my translator was Simeon Kenfak, who could translate into French for me. And when I asked all these people at the beginning, what do people in the villages think cause waterborne disease? And the answer was witchcraft. You have these myths that they don't know about the germ theory of disease. But after the workshop then, that everybody was given then PML materials, they understand. They were really good about that, understanding that you could add science to things. Now, I'd like to show you then about this project, a strategy to eliminate waterborne disease in Lower Nakach, Kenya. The goal is 0 waterborne diseases. And so this is with the Friends of the Old Project in Lower Nakach, Kenya. Okay, we call it PHOTO. And Africa then, this is the equator. It runs right through Kenya. And this project then, this is Nairobi, but it's up here near Kisumu. And we take a closer look. And this is the project that has in Katito town. That's where its headquarters is. And so this is an area that has 70,000 people. And my wife and I were there before COVID every year for 10 years. And these are the women that run the project. These are the women that, well, and this is a village there. This is a house. It has no electricity. It has no running water. These are the poorest people in the planet. And these are old people like me, but they don't have a lot of resources like I have. And these are going to help, but everybody to help that. And it's the International Water and Health Alliances that I co-founded that funds the project then. And the main drinking water sources in Lower Nakach are rivers, ponds, and shallow wells. So here are people collecting water. Some are going to take it back home. They're also washing there. And how many of you would like to drink that water that she's taking home there without doing anything to it? You probably know, but they have no idea. It's pretty clear water. And how about these kids collecting in jerry cans to take water home? That also looks like suspicious water, but they have no idea about it and the shallow well, okay, it's nice and clear water, but take a look at the results overnight and look at all the E. coli there, a very high disease risk that people have no idea the risk is there. And the Constitution of Kenya in 2010, the Bill of Rights then, it says that every Kenyan has the right to clean and safe water in adequate quantities. Again, empty words. What government official is doing anything about that since 2010? And the main goal of the PHOTO is to eliminate waterborne diseases and to be an example to the rest of the world of what can be accomplished by a community-based organization, low cost, to improve community health. And the strategy PHOTO uses is testing, teaching in the community, and then treating the water. So this is where the PHOTO staff then, okay, and all right, so here's the PHOTO staff, and they've collected lots of water sources from all over the 13 locations. And I go through the testing methods, and I also go through how bacteria grow, okay, and how we're looking for an indicator. And then I also explain about the coli alert test and the Petrifilm test and how the results will be for that. All right, now you're microbiologists. Go ahead and inoculate coli alert. Go and you're ready to incubate those, inoculate Petrifilms. The next day we got the results, and you can see there are only a couple that are clear. All the rest of them are yellow, and most of them will fluoresce. And take a look at all the water sources that they have from the rivers and the streams, very high disease risk that they've been able to demonstrate. And also from the ponds, very high disease risk. And so Mary Beth and I then have brought the PML materials, coli alert, Petrifilms, pipettes, WhirlPacks and supplied them all along so that the women could go out to the various locations they are and to the villages and teach people about their water and eliminate waterborne disease from Lower Nakach is the object then. And so the women, 2 women in each of the 13 locations each month has 2 training sessions to go over the water testing of the water before and the water after. And those are bacteria that came from feces. We can't drink that water without testing, without treating it. And how to empower people that had no background. How proud she is that she can now do water testing, just like a university professor could. Disinfection options, well, chlorine. And if we take a look at rates in the United States of typhoid, that used to be a lot per 100,000, but then chlorine was introduced in 1908 and it got to be very big. And then by 1941, it was gone. Treat water with chlorine, that'll get rid of waterborne diseases, all right? And so in Kenya, they have the chlorine solutions, these AquaGuard, that, this is the AquaGuard chlorine. It's 1.2% sodium hypochlorite. One capful will disinfect 20 liters, and a bottle will disinfect then 1,000 liters. So these are the jerry cans, 20 liters that they have there. And then if you do the testing before and after of AquaGuard testing, treatment, you'll find, oh my gosh, that convinces people that they have to treat their water. And each month we have over 9,000 AquaGuard bottles that are distributed to the households and to the schools. Another disinfection is solar disinfection. And being a microbiologist, knowing food microbiology, that milk pasteurization is 71 Celsius for 15 seconds, we know you don't have to boil milk. You don't have to boil water either, because the microbes that cause disease in water, they also are susceptible to heat. And so we could pasteurize water as we pasteurize heat, as we pasteurize milk. And at my university, Cal State Sacramento, I had students that formed the background of these testing, and heating water to 65 Celsius would pasteurize the water. And we published this, when was that? That was 1984. And how can one verify that water has been heated to there without a temperature? And that's where we developed this water pasteurization indicator. It's a tube with a wax that melts at 65 degrees Celsius, okay? And this is the WAPI that we call it. And you put this in water, and if it heats enough so that the wax melts and falls to the bottom, you know you've gotten to pasteurization temperatures then. Okay, and so the WAPI wax melts at 65. That's going to kill all the other germs that are there. And a student and I, we published on this in 1999, all right? And so you can do solar pasteurization. This is a simple cook it solar cooker that can cook food, but also can heat water to pasteurize water. And when the WAPI wax is melted, as it is here, and we do the tests before and we do the tests afterwards, people can understand that pasteurization would work. You don't need to boil water. And the other thing, I had a workshop then for the Ministry of Health in Kenya. And my friends then with the photo project, okay, they were solar cooking while I was leading the workshop. And they had food in each one of those that they're cooking in the simple cook it. And afterwards, then they could serve lunch, solar cooked lunch to the participants. And all of that food could be cooked in 3 hours with sunshine. So that's another thing outside of water, but people that only know fire cooking, that cook it could also help them out. Now, there was a cholera outbreak in Kenya in 15 and 16, lots of cases in the county that Lower Nacotch's in and other counties, but zero in Lower Nakach, Kenya. And then one of my former undergraduate students did his PhD work on the photo project. And he had a team that went collecting, and they showed then that after 377 households, 95% treats their water each and every time. 96% had low disease risk stored water by microbiology testing. Evidence-based microbiology at the community level changes behavior. And there was a video presentation at an IDEX thing. They call it the miracle at Lower Nakach, but it's really good science, good microbiology. And the IDEX company that makes Coli Alert then awarded the Friends of the Old this Water Humanitarian Award that got to then the photo people and how delighted they were to be recognized by the company that makes the best water test in the world. Why then is not the Coli Alert and Petri film tests used worldwide? Well, there are these UN sustainable goals and that were implemented from 1950 through 2015 through 2030. And they have this fairy tale goal. By 2030, achieve universal and equitable access to safe and affordable drinking water for all on the premises. There are 274 villages in Lower Nakach. And to think that they're going to get things, that's absurd that they're going to, but that's what the water community does. And then the Joint Monitoring Program appears at World Water Week in Stockholm every year, and the state of the drinking water and how far behind the world is of achieving SDG 6. But they also tell countries the only way you can test water is if you do obsolete, expensive membrane filtration testing, and that you filter 100 mil. You've seen with the Petri films, 1 mil has got a lot of E. coli in it. It's absurd that unfortunately the Joint Monitoring Program is promoting this quack microbiology to countries around the world. And the goal is SDG 6.1, which is not going to be there. But I really like the people with HIV/AIDS, with malaria and tuberculosis. They want to get to 0. So that's what the goal should be, 0 waterborne disease. And you as microbiologists then, that this empty resolution that they've passed, but you as a microbiologist, you could do things. You could, in your countries then, bring water testing to the community, raise awareness among politicians that water's unsafe, make them get, have them do things. And so that's where I would like you to, if you're interested, I would like you to connect with me. And my email address is rmetcalf@csus.edu. But, let's get people, get them the Coli Alert and the Petri film tests, and get them chlorine, either this or as tablets like this. Let them know about the let them know about the solar cooker, the simple solar cooker, and then also get them this booklet, and you can do something. And that's where I encourage all of you to become champions for those 2 billion people that don't have safe drinking water. And if you would like to get in touch with me, I'll see if I can try to get you a Coli Alert, some of the testing materials and a flash drive with teaching materials in it so that you could educate the others microbiologists there where you are, and you could perhaps plan for a project that would start educating people about waterborne disease, looking for E. coli, how to do it, and then demand that your government gives, some type of chlorine to the people there to get rid of waterborne disease. So I'm hoping that a number of you will be interested, and if you are interested in emailing me and describing the situations in your country where waterborne disease plagues the people, but the photo project strategy could address that. Africans, teaching Africans and teaching those in Asia and Latin America then that this is the way to go about it. Testing, teaching, and treating, mainly with chlorine, but also with the solar option with the Cookit and the WAPI. So I will then end my presentation here, and I'll be glad to answer questions that you have.
Thank you, Dr. Metcalf. We had great participation. You had a lot of virtual round of applause that came through Zoom and a lot of comments. I think you're definitely going to hear from our ambassadors after this. There's definitely a lot of interest in further connecting with you. I wanted to ask you a couple questions. So we have the pleasure of having both our country ambassador and young ambassador to Ukraine on the call today. And they were just wondering to get your interest and sort of your feedback on if you think this type of testing could possibly be done in combat situations. We have, challenging times throughout the world, and there's definitely a lot of need for this type of testing all over the place.
I think it would be very useful here. If people had a PML and they're thinking about a well or a stream that they're getting water from, they could easily test it and to see if it would be safe or not. And then give them some type of chlorine if it's not safe to treat the water with. I assume water sources you haven't ever tested might be contaminated, but this could give them evidence. This could give them the science behind it, and easily they could learn about this because you, as a microbiologist, could Zoom with them, as I could Zoom with anybody around the world and anywhere you, and go through the testing and a little bit of techniques to do that, and then bring it out into the field. And so for our beleaguered friends in Ukraine, that most of the United States citizens want our government to help and are disappointed that that's not getting there, but maybe with a microbiology you could do that. And also the water testing for treatment plants. You have to do 100 mil tests all the time, very likely in Kyiv or other places. But, if you had a lot of these 10 mil tests, you're going to get the same results. If the water's been treated, it's going to be clear after treating. And with this 10 mil test, you don't need incubators, then you can put it on your body and make sure it's still clear. If it turns yellow, you might have a problem. But instead of doing 100 mil tests, you could do this 10 mil tests to do that. So that would be another option that I have.
No, I think
Thank you a lot for your response. Yes, thank you so much, and for this lecture too.
Thank you for your question, Julia. It was really appreciated. I have one other question for you, Dr. Metcalf, before we run up on time. Someone asked about sort of the detection limits of the Cholera and the E. coli Petrifilm tests and sort of how reliable it would be, and if you knew anything about the rate of possible false negatives that, or any way the test might underperform.
They've done a lot. These tests have been vetted, and there are countries around the world that accept Coli Alert in their countries because they've done a lot of tests in the 1980s about that. And also the Petrifilms are accepted around the world to do that. And now you have to understand what the limit for Coli Alert. If you have one E. coli in a 10 mil sample, it will give you a positive test. It's a presence or absence. Whether you have one E. coli or 100 E. coli, you get a positive test. Now, bacteria are particles, and on a Petri film, if you get 10 E. coli colonies, if you set up multiple Petri films, quite often because they're particles, you might have about a 30% variation. And so you might have a 10, you might have a 7, you might have a 13, but what you do know, you got E. coli there. It's either a high risk or very high risk. And so there is variation, just like membrane filtration would give you variation because bacteria are particles. It's about 30%, but that would be within the limits. You really want to know, is it a high disease risk, very high disease risk, or not? And then you take a look at the 10 mil coli alert test, and is it positive or negative then for E. coli then? Is it not a low disease risk, someplace between 0 and 10 E. coli per mil, or is it a low disease risk?
Absolutely. I think this is really great. I want to encourage all of our ambassadors, and you're welcome if you didn't get a chance to write down Dr. Metcalf's email address. Please feel free to reach out to me as well. I'm happy to make that connection. And there was one question here at the end asking if this could be a project proposal. And for any of our ambassadors on the call, I do highly encourage you to reach out to Dr. Metcalf directly, get more information, get some of those training materials that he mentioned on his flash drive, and consider doing something about water testing in your local communities for next year's Ambassador Project Fund call for proposals. We definitely very much support that idea. And I think this was a wonderful workshop. So thank you so much for your time.
Okay, and thank you for arranging this. And just as a reminder, who speaks for the 2 billion people that don't have safe drinking water? You're a microbiologist, and I want you to be a champion for those people too, because they are voiceless and they are not represented. And nobody talked about waterborne disease at ASM Microbe 2025 in Los Angeles. But we need to get, as microbiologists, we know how to address this, test, treat with chlorine, educate the community. So let's get busy with it. And I look forward to anybody that's interested to connecting with me, and let's be champions for the voiceless 2 billion out there.
Perfectly said. Thank you guys again. I'm so happy we had such great participation today, and we look forward to following up on this. Thank you so much.